This presentation is entitled Yeast Complementation, and it's presented by myself, Lindsey. We'll first begin with a brief history of complementation testing and what we can learn from it. Edward B. Lewis first developed complementation testing as a test of gene function, where two genotypes with recessive alleles are combined by a cross to test whether the genotype of one parent can supply the function absent in the genotype of the other parent. For his work in complementation testing, Edward B. Lewis won the Nobel Prize in Physiology or Medicine in 1995, before his death in July of 2004. The results of complementation testing show us the location of mutations on certain genes. If two mutations don't complement, the mutations are on the same gene. If two mutations do complement, they're on different genes. It's important to note that this only works for recessive mutations. In regards to yeast, yeast was first studied by Louis Pasteur. He found that alcoholic fermentation required a living organism, which he discovered to be yeast. Yeast can be either haploid or diploid and has two stable mating types, types A and type alpha. Yeast is popular for study in complementation testing as it's a unicellular eukaryote. Much of its biology is conserved in other eukaryotes, including humans, and it's also very easily isolated. The complementation test, when conducted with accuracy and precision, yields two highly identifiable pink or cream results. The pink color indicates complementation, meaning mutations are on separate genes. The white or cream color indicates no complementation, meaning mutations are on the same gene. The pink cells indicate no adenine is present, and their distinctive reddish-pink color comes from aerobic oxidation that occurs as these cells grow. For our experiment, we'll cross alpha and A strains in an agar plate, and while we have no real way of hypothesizing which strains will complement one another, from what we know about color as an indicator for complementation, we will expect some crosses to be pink, indicating complementation, and some to be a cream or whitish color, indicating no complementation. We also hope to complete clean and precise crosses, such that there is no cross-contamination or smearing resulting in colors other than pink or white. For our experiment, we'll be crossing strains A through C with strains E and F, with the following crosses to be conducted listed under Step 1, Crosses Conducted. The next and perhaps most important step is mapping our agar plate in order to precisely track the results of our crosses. Our map is listed under Step 2, Map of Crosses. We then used loop spreaders to collect drops of each sample in the agar plate. We sanitized the loop spreader with a heat gun and then placed a drop of the strain to be crossed gently on top of the sample in the plate. We made sure to sanitize the loop spreader thoroughly with the heat gun in between every sample collection in order to prevent cross-contamination. We then allowed the plate to incubate for 48 hours. The results of our complementation tests were quite clear and precise, indicating no cross-contamination but for a small amount of yeast that was spread between the first two rows. We see no complementation between strains A and E and strains A and F, indicating that those mutant strains are located on the same gene. We do see complementation on all of our other crosses, indicating those mutant strains are located on different genes. Fortunately, our results were very clear and unambiguous. In conclusion, we were able to see clear complementation between 4 of our 6 crosses and a clear lack of complementation in 2 of our crosses. It's important to note the necessity of precision when performing strain crosses. While none of ours experienced cross-contamination, we can see a small amount of yeast, as indicated by the red circle on the slide, from our AE cross, came very close to contaminating our BE cross in the second row. Had that occurred, we may have seen a mixed or inaccurate result, which may have indicated a lack of complementation in the BE cross, as there was no complementation in the AE cross. However, we learned when a complementation test is performed with accuracy and precision in order to prevent cross-contamination, it's an excellent way to delineate the location of mutant strains on genes.